mild, sometimes quite severe. So there is also this phenotypic variability, as you can see in PXE patients, that is also represented in these fish, and gives opportunities to study the factors that may contribute to this variability. And then if you have a closer look at their tissues and their skeleton, you see that the PXE zebrafish has a body and a tail that is completely deformed compared to the straight tail of a normal zebrafish. And if you really look very close, then you can see that there are clumps of mineralization, extra mineralization, all around these tissues in the zebrafish, representing the ectopic mineralization, as you can see in PXE. And on top of that, there are also other bony abnormalities like the ribs over here that have this bifid ends that are shorter also than in a normal fish. You can stain this with a calcification stain and use that to quantify it and then really put a number on this fish has this much more mineralization, the other fish has a little less mineralization. So not just the observation of I see something and it's different between two fish, but you can really put a number on it so it's quantifiable. And you can also put them in a CT scan. So you can put a lot of fish in the CT scan because they are really very small. And then you can do this live, so without having to euthanize them. And then you can see that extra calcification will color up blue on a CT scan, and all the bluish aspects you see all around is extra mineralization shouldn't be there. And this is just a detail showing these really irregular clumps of extra mineralization that you can see. This is very nice. So what you can do with that, or what we do with it, is we try to answer the big questions that we have in PXE. And those questions are, first of all, treatment options. You know that there are a lot of indications of potential treatments. There are also issues about side effects, the efficacy of these treatments on long term, on short term. So there's certainly a need to broaden that and pick out as many potential compound screen or treatments that we can have, and then just choose the best ones, which may not be the same for everybody. And also specifically look at the genetic aspects, and certainly to try and prove that a certain modifier gene, what we talked about yesterday, is actually modifying the phenotype. And just to show you how that works for compound screening, we have a number of zebrafish that are exposed to what is called a compound library. And basically this is a collection of, for example, 1,000 different drugs. And we expose the zebrafish to these drugs. And then we look at the amount of calcification using this quantification. And see if they are able, one of these compounds is able to prevent, for example, calcification. But we can do the same with adult fish who are already heavily calcified, to also look at, are some of these compounds able to reverse the calcification, which is, in fact, for patients probably much more interesting, because if you're a patient, you already have the calcification. And this will help us to identify among a lot of compounds the most interesting ones in a very rapid and cheap way. But that is just the first step. And then you have to go to what are the side effects. We have to go to mouse modeling before entering in later stages the clinical trial. But if you have to do this in a mouse model, for example, this will take a lot of time and a lot of money to screen thousands of these drugs. So zebrafish are an ideal system for that. And the other thing, finally, is for the modifier genes, the principle is very easy. We use a PXE zebrafish, and then we just inactivate the suspected modifier gene. And we look at the calcification. If it calcifies more, that means that the modifier gene is actually modifying the disease. It increases the mineralization, and it is a first step in validating such a gene. So in conclusion, zebrafish have a highly similar genome compared to humans and can be easily edited. ABCC6A is the homologue of ABCC6 and gives you hypermineralization symptoms that are easily quantifiable and that you can use to search for, or that we use to search for novel drugs and to identify modifier genes. Although I have to admit that for the latter, we are slowly conversing to CRISPR modified cell cultures to reduce the amount of animals that we have to use in our research. So thank you very much. And I'm happy to address any questions.
Thank you very much, Olivier. Could people either put your hand up or put your question in the chat? Or if neither of those work, then you can unmute yourself. Are there any questions? Wow, you gave such a good element.
So clear.
Explanation. Yes, very good. One thing I will just mention that that we got a couple questions in chat just to me, and maybe you can make a comment, is that often people, just regular old people, say the PXE gene and think that people and animals don't have that gene if they don't have PXE. So maybe you could mention again how it is that you've altered a gene that already exists and maybe make that point.
Yeah, that's something that's a mistake that is often made, and sometimes we make it ourselves to make it a bit comprehensive, but there's no such thing as a breast cancer gene or a PXE gene. The basis is that all the genes, we all have these genes, and as I showed you today, the reason why you have a disease is when there is a genetic defect that occurs in that gene. But if it works normally, then you will not get it. So it's the genetic defect that is specific for the disease, for getting the disease, not the fact that the gene is present, because we all have them. And as you heard yesterday, we all have them twice because we have most of our genetic material in double. So really what we do is the gene is there in these animals, also in these cells. We can make PXE cells. But what we do is we introduce a genetic defect. So we make sure that with CRISPR that the gene doesn't work normal anymore and that is not able anymore to produce a normal protein, a normal ABCC6 transporter. And the example that I showed here is the most drastic modification. We just cut it in two and then it doesn't work anymore. So it doesn't produce any protein. But it's more difficult, but we can also try and introduce a specific genetic defect like a missense mutation that we talked about yesterday, which can have a much more subtle effect. So not the protein isn't there anymore, but just the protein, it is there, but it works not as drastic, it doesn't work as optimal as it should be. So that is what CRISPR tries to do.
Great, thank you so much. And thanks for staying on time.
You're welcome, you're welcome.
Next we will